Hi everybody, this is my first live appearance on Facebook, and I want to see if I can start using that medium to communicate with you all and share in a better way about my investigations on intelligent design. So, as probably many of you know, I am writing on a book. Its name is Origins: What Cause Explains Best Our Existence and Why. And the main objective is to investigate molecular biology and what happens inside cells, their origins, how can their origin best be explained by evolution or design, and do this in a very detailed manner. That means going and dig as deep as possible, because as Behe said once, origins can best be explained by looking into molecular biology. In the molecular world, we see the real mechanisms which drive life. So there we can find the answers about origins. That's the best way to explore that. And I am right now investigating how genes are expressed, and the method is the gene regulatory network, which I have posted a few things about in the last few days, and it's amazing. I mean, what you see when you start digging there is just incredible complexity and design in every level. And what is interesting is that you have a chain of interconnected factories in the cell, and that begins with the chromosomes, which need to be expressed at the right moment and at the right place. So this network needs to find the genes, and in the human cell, that is in a string of chromosome-sized 3 billion nucleotides. That's a huge number. So how does the cell do that? Actually, that is something that is being investigated by science. Science does not have all the answers because it's incredibly complex. But what can be said is as follows: that the gene regulatory network has three main jobs. That is developing an organism, a multicellular organism. Then it needs to respond to environmental stimuli and adapt to new food sources. So it has to cure with three different things at the same time. And how does it do it? That's how I say it. There are many mechanisms that regulate the genes, how they are found and expressed at the right time. And what I've seen in the last few days is that histones, which wind up DNA around them, they have a tail which is like a hard disk. It's a string of amino acids upon which the writers that are protein writers, they are like a pen on paper. You can imagine the tail like a piece of paper, and these proteins, these writer proteins like a pen, they write on that tail, and there is a language which they use, which science doesn't know exactly what kind of language it is, but it is a language, and that's incredible. I mean, not only do these proteins write on single histone tails, but each how can I say, each nucleosome has 8 histone proteins, and these proteins, they have each one of them a tail which protrudes out of the nucleosome, and these proteins, there are 8 tails that protrude out of the nucleosome, and these proteins, they can read one tail, they can write on one tail, but they can also interact amongst these 8 histone tails. And there are about 30 million histones in a human cell. And these proteins, these reader proteins and the writer proteins, they can interact one with each other. And that's just amazing. And science is just now beginning to explore that language and how they do it. And this brings the complexity of gene expression to such a high level. I guess that it will take decades for science to find out how that actually works. And it's evident if you if there is a mutation in that regulation, in that network, then genes will be expressed in a different way, and what will be the consequence? Cancer. That means that if this whole thing is not fully set up right from the beginning, then it can't work. So that raises the question, how did it come to be? How did it develop? And it seems to me that a gradual fashion, like evolution, can't set it up. I mean, that is a language that has to be set up, upon which different protein readers can communicate with each other. And languages come from intelligence. Languages cannot just evolve. And languages are not something physical. Language is something metaphysical. So this seems to me amazing evidence of design, you know. And so let's suppose you have all this kind of information exchange set up. You need the histone, the histones, the core, the nucleosome. Then you have the DNA wind up on the nucleosomes. And you have all the machinery which can read the messages on the histone tails, which inform the other machinery of the transcription factory. Look, here, that section of DNA has to be expressed. So there is another level of information exchange. From the knowledge of proteins when they read the histone tails, oh, this section of DNA must be expressed, then that information goes to the RNA polymerase transcription machine, which knows, okay, that section of DNA must be expressed. So there is another machinery in place that are transcription factors, which check on the DNA string where is the beginning of transcription. And when that place is found, then a huge new different armada of proteins, they bind to that beginning section of the DNA and start transcription, and the DNA string is transcribed. And they transcribe until a new sign comes which says stop. Here the gene section is finished to be, it's that string that needs to be transcribed, and then the machinery stops to transcribe. And then you have a messenger RNA string which goes through further post-translational modification. It has to be kept, it has to be brought into the right format, and then it will leave the nucleosome where all this kind of stuff happens, and it is transported to the ribosome, which is the translation factory. So you have here three interdependent things. You have the gene expression mechanisms, you have the DNA transcription factory, then you have the product of all this, which is messenger RNA. That's a string of information which travels out of the core of the cell and goes into the cytosol to the ribosomes, and there it will be translated and form proteins. And when the protein string comes out of the ribosome, it is tagged and it will be addressed, and it will go through the another factory which will do further work on the protein. It will fold into 3D form, so there is a transformation from a messenger RNA string, which is a digital form of information, and the ribosome transforms it into a three-dimensional protein, which is analog information. Okay, and usually these proteins, they are very complex. There is not just one amino acid string which folds, but there are 3 or 4. They have different levels of complexity. There is cofactors that have to be inserted into the protein. So this is another part of another factory which produces proteins. And when the protein is ready, then it is tagged, it is put into mail, like into a post office, and then it is transported to the place where the protein will begin its function. And that transport is also amazing. And sometimes in eukaryotic cells there are tubulins which have also on them. It's like it's a tubulin code. It's another code which directs the protein through other kind of proteins which transport these proteins, and it goes to the final destination. So you have here various factories which are interdependent, and one of these is missing, then nothing goes. So if you have all these kind of factories and machineries interconnected, but if just one thing is missing, then proteins cannot be synthesized. So if you have, for example, inside the ribosomes tRNA, which is the translation mechanism, then if you don't have that RNA, nothing goes. So what can be said about this? You have a cell, eukaryotic cells, which have various interconnected factories which have to work together, and they have all the pieces need to be interconnected together. It's like a production line. If where one robot is missing, nothing goes. The whole factories and the whole production line ceases to work. And this is extraordinary evidence of design. I mean, evolutionary mechanisms just don't explain such things, and Behe's concept of irreducible complexity is more and more evidenced through all these kind of things that I described now. So This is amazing, and I am trying to collect this information in as much as detailed fashion in order that it can be appreciated and make our faith in design, in creation, stronger. Thank you very much.